The last section of the organic molecules chart is on nucleic acids. They are composed of carbon, hydrogen, oxygen, nitrogen, as well as phosphorus. As well, they are the largest molecules in the body. Nucleic acid polymers are made up of monomers, which are called nucleotides, and they contain a nitrogen base, a pentose sugar, which is either going to be deoxyribose or ribose, as well as a phosphate group. And included in this are also ATP, ADP, and AMP. So the two largest nucleic acids are deoxyribose DNA and ribonucleic acid. Back when I was in grad school, it was really funny. They actually had a beer called deoxyribonucleic acids, which we thought, being the big nerds we are, was really funny at the time. As you know, DNA holds the genetic material for synthesis of proteins. So it's the blueprint of the cell. It's a double-stranded helical molecule, a double helix. So if you think back to the alpha helix that you learned in the about in proteins, it's similar to that. These nucleotides contain a deoxyribose sugar for DNA, a phosphate group, and one of four nitrogen bases. These bases come in two different groups. There's what are called purines, adenine, and guanine. And there is cytosine and thymine. The bonding of the nitrogen base from one strand to the opposite is very, very specific, and it involves hydrogen bonds, just like we saw within proteins. And remember, again, these hydrogen bonds are the weakest type of bond. So it's important for your exam that you know the difference between the different types of bonds. So it follows complementary base pairing rules. And what this means is that adenine always pairs with thymine and guanine always pairs with cytosine. There's lots of ways to remember this pairing. Memory tricks or mnemonics will help you a lot in this class. When I first learned about this, I was going to college in North Carolina, and there's two different colleges in Greensboro. There is one called North Carolina A&T, so that's how I remembered it. And there's another one called Greensboro College. And that's how I always remember it. Another way that you could remember this is possibly remembering Grand Canyon. But again, there's a lot of different ones. It doesn't matter what the memory trick is. The important thing is it sticks out to you and helps you remember it. So this is our structure of DNA. And again, there's no diagrams on the test, but you should be able to identify the different parts of it. And again, you don't need to know the structures. But when we look at a nucleotide here, again, the three parts of it are a phosphate, a sugar, which in this case is deoxyribose, and then a base, adenine or thymine, which always pair up together. In this case, there are two hydrogen bonds between adenine and thymine. In the case of guanine and cytosine, there are actually three hydrogen bonds, which really should be drawn with just dashes because they're really just weak attractions, as you have learned by now. But you can see this three-dimensional shape, and it's these specific bases which are going to eventually produce amino acids and build the protein. Another nucleic acid is the RNA, which links to protein synthesis. It's a little different than DNA because it's single-stranded and it has ribose instead of deoxyribose. The other important thing is that thymine is replaced with uracil, and there's three different varieties of RNA. There's messenger RNA, transfer RNA, as well as ribosomal RNA. So the messenger RNA is simply a copy of the DNA that's made during what's called transcription, which you'll be learning more about in chapter 3. Transfer RNA is what's going to basically what I like to think of as taxi in the corresponding amino acid. And then ribosomal RNA is what's actually located in the ribosome and also specifically in the nucleus also. So it's more of a structural type of RNA in that it doesn't carry out a specific function. Another nucleic acid, which is super, super important, is our energy currency of the cell. So whenever anything has to happen in the cell, it has to have a form of money that it uses for things to happen. And that money happens to be ATP. ATP stands for adenosine triphosphate. And one super important part of this molecule is the prefix, in this case, of tri, because that phosphate bond represents a high amount of energy that can be used for all sorts of things. It powers chemical reactions in cells, offers immediate usable energy needed by body cells, and the ATP has 3 phosphates. So it has 1 adenine phosphate with 2 additional phosphate groups. And that last phosphate is transferred to other compounds used for some sort of work. So the loss of a phosphate converts ATP to ADP, and the loss of a second one goes from ADP to ANP. So we would see a chemical reaction sort of like this. So ATP, when it breaks apart, it produces ADP plus an inorganic phosphate. This is enough energy to power something. When we look at the molecule here, again, we see these high energy phosphate bonds that are hydrolyzed. So water is added to release energy. So it has the base. Again, you don't need another structure. It has the ribose, which is a monosaccharide. So you should know that, as well as the phosphate. So the ATP is with 3 phosphates. ADP is with 2 phosphates. AMP is with 1 phosphate. And then the phosphate can be re-added to then be used again to recycle essentially for another energy reaction to happen. These are 3 important examples of chemical work in the body. In the next chapter, we're going to start talking about these membrane proteins. So membrane proteins are kind of like, I like to think of them like doors or channels that open and close. And in this case, for this solute to actually enter the cell, it needs energy. So this phosphate that we see right here is broken off and released. So we go from ATP to ADP. That supplies enough energy for that channel to open so that the solute or the ion in this case can cross the cell membrane. The next example, which we'll learn about in chapter 9, is mechanical work, where the ATP is going to phosphorylate, and it basically looks very similar in that ATP is broken down to ADP. But what happens is this myosin molecule, which is an important protein, physically moves to bind with its binding site on actin. So the myosin changes shape, pulling another contractile protein, which is actin. And the final example is classical chemical work. So it drives an energy absorbing chemical reaction, which remember in this case is endergonic, since it absorbs chemical energy. So here the ATP is broken down to ADP, And these two reactants combine to produce could be a disaccharide, could be a dipeptide, etc.